at all these words, right? Like these are a lot of words. So we're not going to go over all of them today, but this is here as a resource for you. And I mentioned last stream, if you were able to catch it, a great study strategy is to just write down one word at a time on a piece of paper. So I'd write the word chloroplast, right? And with no notes and no book and no internet, I would just write or draw everything I know about a chloroplast. And you might surprise yourself how much you know about the chloroplast, right? You might all of a sudden start talking about not just the structure of chloroplast, but its role in photosynthesis and its role in, the fact that it was used to be a prokaryotic cell. You might surprise yourself, but you'll also identify in what areas you're confused. So really, this is a huge study strategy. I used it all through college. It was a wonderful way, instead of just looking through flashcards of vocab, it's a process of, hey, what do I know and what don't I know? Because it's a huge thing that's going to separate you from someone who maybe, isn't going to do as well on the test, right? Because you guys are all going to do amazing because you're here, right? You're here. You're spending your Saturday afternoon studying AP biology. So I'm so impressed that you're all here. Okay. This whole unit's about cells. We've got, if we think about characteristics of life, right? Cells are a very big characteristic of living things, right? If we don't have a cell, you're not a living thing. And just knowing its place in, if you have to refocus yourself sometimes, like, I don't know, when I was younger and I'd be studying something complex, I'd be so hyper-focused on what I was studying that I would forget the big picture. So I just encourage you not to forget the big picture, that, this is biology. We're learning about life. And why are we learning about cells? Well, because all things are made up of cells, and all cells need to do stuff. They all need to take in things. They need to get rid of things. They need to have transportive materials, like even gas, right? So we have oxygen in our lungs, oxygen out. Everything has a purpose, and everything has a cool structure that allows it to do its purpose. So every now and then, if you're ever feeling like way too overwhelmed with the biology, zoom out and be like, all right, what's the bigger picture? Why is this important? All right. Lots of different cells you should know about already, like a difference between a prokaryotic cell. So here's a typical prokaryotic cell. Remember, prokarya means there's no nucleus and no membrane-bound organelles. So you've got, and I'm not going to tell you too much because I've got a little quiz question for you coming up. Then we have eukaryotic cells. This is just a picture of a plant cell. If you were to look closely, you'd be able to know it was a plant because you could see the chloroplast, you could see that there's a cell wall. And so these are just things that most students learn in their very first biology class. If you took it as a freshman or maybe in 8th grade, those are things you would have already learned. If not, don't panic. But those are just some things that they're not too difficult to remember or to learn. So a lot of kids come in already knowing that. So here are some poll questions that I'm going to have for you. So give me a second. I'm going to pull this up on a poll. Okay. So which organelle do all living things contain? So which organelle do all living things contain? Keyword is organelle. Organelle, organelle. I don't know if this lets you vote twice or if it just gives you one chance to vote. But we're looking at which organelle do all living things contain? And my hint word was organelle, not macromolecule, if that gives you any indication. All right, let's see how we do. So this one's a little tricky because of the keyword organelle. A lot of people said DNA, but the correct answer is ribosome. Ribosome is an organelle. An organelle, by definition, is a subcellular component. If we remember back to Unit 1, DNA is a macromolecule, not an organelle. So it's a little different. I'm sure there's some scientist out there somewhere who would fight me on it and say that like DNA is an organelle. But what I was wanting for you to see here is not just, the fact that ribosomes are present in everything, but how ribosomes being present in everything are evidence for the connectivity between all living things. And so I know I'm jumping ahead a little bit and having you think about evolution for a second, but this is something important to lay the groundwork for now, that if we're comparing things and saying like, wow, everything has a ribosome, like that's a highly unchanged structure. How cool. They all have the same thing. Continue to think about biology through that lens of what do these things all have in common, right? Like when we jump ahead to cellular respiration, all living things can do glycolysis. It's amazing, right? Because you don't need a mitochondria for it. It happens out in the cytoplasm. So just kind of keep trucking with that thought of how is this showing the connectivity of living things and how it's not changed. Okay, so really good job on that one. All right, this one is also a poll. So let me pull it up. Show poll. All right, so the ribosome, chloroplast, and mitochondria all have something in common. And I know my things are kind of cut off, but hopefully you can still read it. But it's A, do they contain nucleic acids? B, they're found in prokaryotic cells. C, they're found in animal cells. Or E, none of the above. Oh, shoot. My poll is wrong. Sorry. It shouldn't say found in eukarya. It should say found in animal cells. So oh, shoot. Yeah, it's not supposed to say found in eukarya. It's supposed to say found in animal cells. So option C is wrong. I will just tell you that. Option C is wrong. I'll have to go back and take that out. Sorry, everyone. Okay, so again, I'm super sorry about this. My poll and my multiple choice were not the same, so I apologize. But if we think about ribosomes, mitochondria, and chloroplast, you are the 28% of you who said they contain nucleic acids. You're right. The answer is A. And so that's just a little factoid that most people kind of rush through, and they don't make that connection that like, hey, look at mitochondria have their own DNA. It's circular, just like a bacteria, and same thing with chloroplast. They have circular DNA as well. And so if you think ahead to your gene technology unit, when you learned about plasmids and how we can take a plasmid and cut it with restriction enzymes and insert it into a bacteria to make it express whatever gene we want. Like a lot of you probably did the pGlow lab where you inserted the jellyfish gene into the bacteria and watch your plates grow. Or if you didn't, you might have done a virtual version of the lab. But either way, this is also kind of showing us that evidence that like, hey, I think, these mitochondria and chloroplasts seem very bacteria-like, right? The same size, they have DNA, they self-replicate, like a lot of interesting commonalities there that lead us to thinking about the endosymbiotic theory. Also with ribosomes contain DNA. There's ribosomal RNA in there. So anyways, just something to think about, and really good job for those of you who caught on to that one, even with my bad, incorrect polling. I apologize for that. Okay, new poll. Hopefully this one's right. We shall see. I'm double checking before I post it. Yes. Okay, so plants have a specialized organelle, a specialized version of this organelle that can absorb large quantities of water. So it's a special organelle that can absorb large quantities of water. It's for plants. That's another keyword. And don't be afraid to be wrong. So these polls are anonymous, so even if it's wrong, it's okay. And remember, when you're wrong, then later on, you'll be like, oh yeah, I remember that one time I was wrong because I answered this and now I remember it's this. So don't be afraid to be wrong. Don't be upset if you're confused either, because it's through that confusion in where we learn the most. But you are all wonderful. Great job. Large vacuole is our answer, right? So large vacuole is our answer. You might have been confused by the contractile vacuole option, but remember that's in our protists that are living in freshwater environments where they're kind of constantly taking in water. So yeah, good job. Let me hide this. I'm already getting better at this tech thing. Okay, large vacuole, wonderful. This one I don't have a poll for because I just want to make sure we're moving along. What type of transport is pictured here? This is a cartoonized version of a blood vessel, or technically a capillary, near an alveoli cell. So that's in your lungs where gas exchange happens. So you can either think it to yourself or you can type in the answer in the chat. It's whatever you're comfortable with. Yay, look at all you answering. I'm checking out the chat and how you're all explaining what a contractile vacuole is. You can actually, there's some pretty good YouTube videos where they show a contractile vacuole of a protist, and they show it filling with water, and they show it contracting and water expelling out of the cell. So it's really cool. You could look it up on YouTube. Excellent. Great job. So, and I love Ellie. She even gave a why, right? So she gave evidence. She justified her answer, which is huge for the AP test. You got to know the answer, but then also maybe be able to justify and say why that is. So awesome. Good. So your answer here is going to be diffusion, right? Which is a nice passive transport. Gas exchange is constantly happening in our bodies. It's also happening in other organisms, right? I like to think of all this from a very human point of view. And that's not like egocentric of me. I just think the human body is incredible. And I think when you think of body systems and how they work, it's all biology, right? So anyways, it's a very applicable, relatable thing when we're talking about body systems. Okay, this type of membrane transport requires energy input. So most students feel really comfortable with this one. Requires energy input. I'm just going to keep flying. It's active transport. The word active makes me think like, oh, it's lots of energy. Here we go. And most people might think facilitated diffusion because they're like, oh, there's a helper. That must take a lot of energy. But facilitated just has to do with the fact that we've got some sort of transmembrane protein present, and it's going to let things in or out very specifically. So no energy needed for facilitated. Cell membrane, let's moving right along. So we're past the organelles, let's talk about the membrane. So this unit's all about the cell. Every cell has a cell membrane. We should, by this point, we should be able to, using our macromolecule knowledge from last unit, identify that we've got phospholipid heads, right? So sorry, phosphate head, lipid, fatty acid tails, and we've got a double membrane, right? Certain types of questions that you could think of for this topic is, like let's say I threw a bunch of phospholipids in water, how would they rearrange? And you could just draw it right in your own head or literally draw it out, but be able to talk out which part of the phospholipid is where, right? That like the phosphate heads are going to be facing the aqueous solution where your fatty acid tails will be hiding together, right? They're facing each other because they're hydrophobic. The heads are hydrophilic. Got a ton of things in a cell membrane. Every cell membrane has unique properties, right? So like these little shapes right here are going to be your cholesterol molecules. So cholesterol allows for a little bit more membrane fluidity. It spreads everybody out. You've got glycoproteins and glycolipids. Those are there for either cell-to-cell attachment, cell communication, cell identification, right? So like even if it helps identify a cell as, hey, we know that cell. It's got this specific glycoprotein on it. You've got peripheral membrane proteins, you've got channel proteins. So that's why we call it the mosaic, the fluid mosaic model of the membrane. But in the most basic sense of what a cell membrane does, it's just giving you two environments. There's an internal and there's an external. And this is something students don't think about much, but this goes for your membrane-bound organelles too, right? So if we're talking about like a vesicle or a lysosome, or anything like that, that's a membrane-bound organelle, they're membrane-bound, so they have this too, right? So they've got phospholipids, a bilayer as well. So that's just something to think about if you haven't yet. Okay, this is not a poll because it was too long. You can see my answers here wouldn't have worked out on a poll. So if we think of the endosymbiotic theory, I kind of previewed it a little bit, a couple slides before. Give you a second to read that. It says A, DNA and nuclei of eukaryotic cells exhibit a symbiotic relationship. B, chloroplasts and mitochondria were once free-living prokaryotic cells. C, membrane-bound organelles evolved from single cellular prokarya that were enveloped by eukaryotic cells. Or D, both B and C. So type your answer in the chat or feel free to just say it to yourself. Alrighty. You guys are doing great. So on the AP test, you probably won't see questions that say like both B and C or A and sometimes B. But yeah, the answer is D. It is B and C. So chloroplasts and mitochondria, the theory is that they were once free-living prokaryotic cells. So they were just on their own, doing their thing. They had their own DNA. They could self-replicate. But then also C kind of ties the theory in a little bit more, that the fact that these membrane-bound organelles like chloroplasts and mitochondria evolved from your single cellular prokarya. Okay, here's just a picture of that. So we've got an aerobic bacteria, this little pink thing, and it gets enveloped by a prokarya. And it's like, hey, awesome. Like this prokaryotic cell, right, can now harvest or can make ATP, right? And so it became very beneficial for both organisms. And then over time, one other prokaryotic cell perhaps enveloped a cyanobacteria, which is now the precursor to a chloroplast. And something important too to distinguish is that you should be aware or at least be able to give evidence as to why mitochondria were the first, or aerobic bacteria, they weren't called mitochondria, but why this was the first engulfing. Because if you think about it, animal cells do not have chloroplasts. Okay, so chloroplasts were enveloped first. Everybody would have chloroplasts, but only plants do. So chloroplasts must have joined in after mitochondria did. Okay, we're gonna move on to topic 2, cell size, because we're already at 24 minutes, and I wanna get to osmosis as well. So cells have to be small. Why do they have to be so small? A lot of it has to do with diffusion, and hopefully you did a lab like this or saw one online, but it's a very classic lab where you get a big cube, a medium cube, and a small cube, and you end up learning that the small cube is just right. Models are ways where we can study biology without actually using the true thing, right? So we can't necessarily all have access to fancy microscopes to be able to see diffusion happening across a membrane. So instead we use something that can model it for us. So this picture is of some beet cubes. So beet they're dark red normally. And then the bleach is the nutrients. So soaked them in some bleach. Bleach makes things white, right? If you ever ruined your clothes or maybe even used bleach for tie-dye, who knows? But it's showing you that for the larger cell, bleach barely got in, a little bit more on the medium cell, and then the smaller cell, that nutrient is able to go through more efficiently. So we know that diffusion is movement from high concentration to low. And based on this model, cells have better diffusion if they're small, right? So what does this have to do with diffusion and cell size, or what does the surface area to volume ratio have to do with diffusion and small size? So if you've ever seen that show, The Tiny House, I imagine it like comparing a house to a mansion, okay? And let's say it is really, really hot in the house, right? So it's super hot for whatever reason, way, way, way too hot. And you need to open up all the windows, okay? This little house is going to be cooled down more efficiently than a huge house, right? Even if the huge house has lots of windows, right? And you can have lots of wind coming in. This house is much smaller, right? Smaller in volume compared to this giant one. So this is just a picture. Again, this is just a way that I thought of it. I encourage you to come up with your own way of understanding surface area to volume ratio, right? Because whatever works for me might not work for you. And real quick, a quick question back up to our eukarya. Yes, thank you, Jed, for answering that question. Yeah, so in that other question, everybody, about, I think one of my answer options was about the eukarya engulfing the prokarya. I should have wrote ancestral eukaryotic cell because it was technically still another prokaryotic cell. So thank you for that question, Allie. Allie said, I thought the prokarya engulfed another prokarya, and that's correct. Because this all happened way before we even had eukaryotic cells. So I was thinking of it in terms of the ancestral eukarya. But anyway, it's hard when you're trying to write questions that can only be a certain number of words long. Otherwise, it'll take up the whole page. Okay, so surface area to volume ratio. If surface area to volume ratio is high, movement of material improves. This thing in bold, if you have to write it a million ways, draw it out, whatever you've got to do, make sure you know this. Surface area to volume ratio should be high, right? And that's either by increasing the surface area, right? You can increase your numerator, or you can decrease your denominator. And there's lots of different cells that do both, or one or the other. So just make sure you know that, because I know students will get all wrapped up in their heads and they're like, well, wait, I think it should be low, because low is always good, right? Like low numbers are always great, but we want a high surface area to volume ratio because you have a lot of surface, a lot of membrane, which lets more things in, right? Think the surface is the membrane, okay? So if we have lots of membrane for things to move in and out of, then hey, we've got really efficient transport. So this arrow right here is about what is just leading you to this question. What happens to surface area ratio as a cell grows? So if a cell is getting bigger and bigger and bigger, what happens to surface area to volume ratio if the cell is just getting really, really, really big? So you can type that in your answer if you want to in the chat box. So if the cell gets bigger and bigger and bigger and bigger, what happens to surface area to volume ratio? Does it increase? Does it decrease? So cells getting bigger, what happens to the ratio? Oh, you are all so wonderful. Good job. Yeah, decreases, right? So as cells grow, this gets smaller. So in your small intestines, your cell has these projections of the plasma membrane called microvilli. And think of how much surface that is increasing, right? It's not just like a rounded edge. Every little section of this is plasma membrane. And so we've got tons of surface. Small intestine, its job is to absorb materials that you've eaten, right? So if you're interested in, we think about like disruptions, that's another big theme in AP Bio, is that like, well, hey, guess what? Someone with celiac disease, which I always use as an example because I have it. Basically, celiac disease is an autoimmune disease where if you ingest gluten, so like things that are in wheat or barley, like if I had pizza, for example, my body will make antibodies, so it'll produce proteins that attack a certain type of cell that's on the small intestine. So it's an autoimmune, it's auto, so it means like my body attacks itself. And this microvilli turns into these these nubs. So they're not beautifully up and down, increased surface area. So you can imagine, like if they said, hey, they give you all this information about celiac disease, they tell you how it affects the microvilli, make a claim or predict what that would do to absorption. So these villi aren't working, there's self-destruction happening, predict the outcome of that. And so you want to say something along the lines of, well, surface area is no longer large, so that could affect the surface area to volume ratio. Or you could just get right to the point and say, if there's that disruption in the specialized structure that's meant for absorption, then I'm pretty confident that absorption is not going to be as great, right? And so sometimes if you're also thinking about like, how am I supposed to know what to predict? Just use your best, use your knowledge, right? Use what you know and be confident in that. It doesn't have to be this beautifully worded thesis statement. You can just say like, hey, my prediction is, don't say the word my prediction is, but you can say, the mutation would decrease absorption. You can keep it really simple and to the point. All right. Other things we see with large surface area are your inner membranes of the mitochondria, right? The inner membranes where the electron transport chain happens, right? More electron transport chain efficiency means more ATP. your red blood cells, right? They're supposed to fill up with oxygen. So we get this little biconcave shape to it, and that also increases surface area. Just even having that little dip gives you just a little bit more surface area to volume ratio so that it can have really great transport of oxygen and CO2. All right, I'm going to fly through membranes and transport because this is something that I think you all can understand without a lot of talking. So for example, this chart is a really nice visual of what can get in the membrane and what cannot. You don't necessarily have to memorize it because be confident in what you know. If it's hydrophobic, it can go through. Well, small and hydrophobic. If it's small, uncharged, polar, it can get through because it's small enough, even though it's polar, if it's really teeny tiny, it should be able to get through. Larger polar molecules can't get through. Ions, these are your charged things, they can't get through. So think of it in terms of what you know to be true, in terms of polarity and your lipid bilayer, how we have hydrophobic center, okay? And also, if you're too large, getting through that will be very difficult. You would need some sort of transport environment. Is it in a hypertonic environment, hypotonic or iso? What? And then ask yourself, where's water going to move? Or you can look at it in a different way. Cell A is hypotonic compared to the environment, or hypertonic compared to the environment. Where will water move? So give yourself practice problems here. Draw diagrams if you have to, but just kind of throw it out there and be like, hey, it's going to be in this environment. What happens? Okay, now the solutes are added. What's going to happen now? And keep doing that because then you'll identify where you're getting stuck. Okay, so focus on the verbiage of the problems. You're comparing two environments, right? You've got the internal of the cell and the external of the cell. Even if what they're telling you seems complicated, draw a picture, write what you need to write, draw little dots to represent solutes. Do what you have to do to set yourself in a good spot to answer these questions, okay? So I'm gonna work with this equation for you for the rest of our time together because I think it's fair game on the test. These equations are given to you, but if you don't know how to use them, it's kind of useless, right? So water potential is represented by this little thing right there. And so don't get up like Stanley, right, from The Office. He's like, yup, see ya. No thanks. So that might be how you're feeling about water potential, but stay with me. I know you can do it. Water potential is calculated by having pressure potential and solute potential. Most of the time, they're going to just give you pressure potential as a number, or they'll tell you that it's in an open container. And this little paragraph here says pressure potential is going to be 0 if you're an open container. And if that's the case, I love when things are equal to 0. If you've ever been in physics and you get to do initial velocity as 0, you're like, yes, more zeros in the equation, less for me to do. Anyway, so that's one part. The second part, if you are solving for solute potential, you have 4 variables, okay? R is given to you as a constant. Molar concentration will either be given to you or you'll have to find it on a graph. And then ionization constant, it's either going to be 1 or 2. If it's sucrose, it's 1, and they tell you that. But if it's salt, for example, we should know by now that NaCl dissolves into 2 materials, and so your 2 ions, and so that would be 2. And I have an example problem coming up next. The only other big thing that students mix up is that the temperature needs to be in Kelvin. So if it gives it to you in Celsius, just make sure you don't forget that. And again, this is given to you, so don't worry. You don't have to memorize how to convert Celsius to Kelvin. But just little details like that, if you get calculator excited and start typing in your numbers, just don't forget, check your units.